This is lesson 9, part 1 of lesson 9. In these lessons, we're going to show you some experiments to do with osmosis, and I know that Mr. Hancock and Mrs. Lees have recorded demonstrations of these as well, so we'll just go over these. So in this first one, our lesson objective is learning to understand how substances move across non-living membranes. And the non-living membrane we're going to be using in the experiment is called visking tubing. Visking tubing arrives to us as what looks like a dried up piece of plastic. And in order for us to use it, we have to soak it in water to soften it. This creates a bag, but the bag is often stuck together and it has a hole at the top and the bottom end of the bag. So we have to soak it in water and rub it between our fingers to get the bag to open. And then we tie a knot in the one end of the bag. What's special about visking tubing is that it's got tiny microscopic holes all over its surface. So it will only allow very small molecules to pass in and out of the visking tubing. So it acts very much as a model for our semi-permeable membrane. In other words, it will only allow some substances in and out. So in this lesson, I would expect everybody to be able to recall the differences between diffusion and osmosis, and to be able to plot points on a graph to show how the height of the liquid changes over time in our special bit of equipment that you'll see in a minute. I'd expect most people to be able to understand how to measure the rate of osmosis using an osmometer. So the osmometer is a bit of kit we're going to be using, and to choose their own scale and label the axes. And then I would expect some, apologies, it's supposed to say some at the top here, to be able to explain the changes that occur in an osmometer using key scientific terminology, and to draw an appropriate line of best fit through our plotted points. So this is what an osmometer looks like, this piece of kit, and you'll see it set up in the practical demonstration video. It's quite tricky to set this up. As I've already explained, you've got to first of all hydrate the visking tubing, put it in water to soften it, and then you've got to tie a knot in the bottom. You've got to fill it with a sugar solution. So Mr. Hancock will have told you what concentration sugar solution, sucrose solution he put inside. Tie a knot around this very fine glass capillary tube. So this has a hole running through it, a little bit like a thermometer, except this is open at the top. And obviously this is held in place with a clamp stand, so that this bag of visking tubing containing sugar is pretty much suspended in a beaker of pure water. So this is a special piece of plastic called visking tubing, as we've said. Here we can see that the water molecules can move from where they're high in concentration in the beaker to where they're lower in concentration in the sucrose solution, and they make the bag swell. And the only way this liquid in the bag can go is up the capillary tube. So we can measure the rate at which this liquid moves up the capillary tube by measuring the height every minute or 2 minutes. The sugar molecules are prevented from leaving the visking tubing bag because they are too big, and the holes in the visking tubing are only small enough to allow water molecules through. There is a YouTube video. I've put it on the link on Moodle as well. So on page 41, this is where this experiment starts in your book. And here we can see our liquid's gone down in our beaker and the solution has risen in the capillary tube. So the first thing you need to do is to label your apparatus. And we have got the capillary tubing here. We can see the hole in it because it's magnified. We've got B is a mark where the initial level of liquid may have been, and we can see it's risen up here. So they've coloured it in red, so it's just a little bit easier for us to see in this diagram. We have C is the visking tubing itself, the bag. D is like a little elastic band to hold it, and that would be tight against the capillary tubing. E is our sucrose solution, 20%, and F is a knot in the capillary at the bottom, not a no, a knot in this diagram. So these are the results that hopefully Mr. Hancock and Mrs. Lees collected for you. So they ran the experiment over a period of 10 minutes. So time was the independent variable. And the height of the liquid in the capillary tube, which was measured in millimetres to make it precise, was the dependent variable, what they measured. So I want you to plot this data as a graph. So to give you a little bit of assistance on your graph paper, so we have a scale along the bottom, along our our x-axis, time in minutes, and it goes up in 2. And then height of the liquid in the capillary tube in millimetres. I've done a scale going up to 24 because that's the maximum, and I've gone every large square is 2 millimetres. And then you plot your points precisely with tiny little crosses in pencil, please. Or you can do a dot with a circle around, but please don't do like a really kind of like dense dot, okay, and colour it in, because that makes your points not so precise. And don't forget to put a heading on your graph. So in your conclusion, you need to be able to explain what your results show you and explain why this is happening using key scientific terms in terms of osmosis. So you need to link your independent and dependent variable. So if I close this PowerPoint a second, I can go to my publisher document. So these are the results that you had. And there's your label of your diagram here. And if we go down in our page, you can see a conclusion. I've put over time, the level of solution in the capillary tubing increases. This is because there are more water particles outside the viscing tubing bag than inside the 20% sucrose solution. So water moves by osmosis down a concentration gradient from high water concentration below into the viscing tubing, which is semi-permeable. As the bag swells, this forces the additional liquid up the capillary tubing, which is why we get this rise. And in terms of what the graph looks like, I've got another diagram of the graph here. I'm just resizing so you can see it. And we've got our scale. So we can see in this one, the line of best fit kind of levels off at the top, because eventually the solution in the bag would reach equilibrium. Okay, it will gain the same amount of water molecules as outside as inside. Okay, so that's our first experiment. This is included in lesson 9. This is a second experiment to show osmosis in living tissue as opposed to a non-living cell membrane. So the organism we're going to use for living tissue is the potato. So you're going to do a trial to show that osmosis really works in living tissue, in potato cells. And to do this, the equipment you're going to need are a few potatoes, a scalpel, a tile to cut them on, something called a cork borer that looks a little bit like an apple corer, but has a particular diameter to it. It's quite sharp on the edge to be able to cut cylinders or tubes of potato. Filter paper and scales, weighing scales to weigh these on. So you're going to start off by cutting 6 potato pieces using the knife, tile and borer. So you keep the same size borer each time to make it a fair test. And we're going to cut our potato cylinders to around about 5 centimetres, which is 50 millimetres. You need to even up all the chips to the same length. And the reason we do this again is to make it a fair test. It's a control variable. We want all of the potato cylinders to have approximately the same number of potato cells in each one. However, there will be subtle differences because we can only cut to a precision of 1 millimetre using our rulers. We then place the chips onto filter paper just to dry up any excess water, and we record the length, which we know hopefully should be 5 centimetres, 50 millimetres, and the weight of each chip. We're then going to take our chips and put them into different sucrose solutions, and the concentrations we're using are 0 molar, 0.2 molar, 0.4 molar, 0.6 molar, 0.8 molar, and 1 molar. So our range is from 0 to 1 molar, and we're going up in intervals of 0.2 molar each time. If we put these into boiling tubes with the same volume of sucrose solution, so we need to make sure the volume is the same in each of these, then we need to ensure that water is not going to evaporate from these containers and end up making the sucrose solution stronger. So we either put a bung on the top, or if it's in a petri dish or a beaker, we might put some cling film on the top to stop the water evaporating. So this work starts on page 45 in your book. So you've got a few questions here about some of the practical aspects that we did, and particularly about the how science works, the investigative skills. So explaining why the potato chips need to be all the same length to make it a fair test, because if some chips have more or less potato cells in them, then they will gain more or less water, and so that will impact on our results. Why do we dry up the excess water? Because the additional mass from the excess water will give us false results. We only want to know the mass of the potato at the start and at the end after the experiment. The purpose of placing the bung, or as I said, cling film on the top of each container, prevents evaporation of water from the solutions and altering their initial concentration. Our independent variable, this is a variable we change, is the concentration of sucrose solution. And we've already described that the range is from 0 to 1 molar, going up in intervals of 0.2 moles. The dependent variable is going to be the change in mass. You will see tiny, tiny subtle changes in length, okay, but they're not as much to be able to actually measure. And control variables, things we keep the same to make it a fair test. We want the same volume of each solution. So Mr. Hancock will have told you in the practical video, the same length of the chip, the same diameter of the chip, so it's cut with the same width cork borer and left for the same amount of time. So at the end of roughly about an hour or so, you remove the potato chips from their container. You dab any excess water onto a filter paper. You don't want to do this for too long because it will literally draw the water out of the cells. You design a table for your results on page 46, so make sure you have clear headers with appropriate units. And then you record the final mass of the chip. So you will have the starting mass of the chip, so hopefully they will all be the same mass, and the final mass. And then you're going to work out a calculation, and this calculation comes up frequently in WJEC exam papers. They like to know whether you can calculate a percentage change. To work out a percentage change for each potato chip, all you need to do is have the change in mass on the top, and this could be an increase or a decrease. So if it's an increase, we show it with a positive sign, a plus, whatever the mass is. And if it's a decrease, it would have a negative sign. And that goes, divide the change in mass by the starting mass, and then times it by 100 to work out the percentage. So once you've worked out the average for your chip, your teacher will give you more data to help you work out class average. So you're just going to do it for one set of data. If we were in the lab, we would be asking each group to collect a set of data, and we would work out a class average. Why? It makes our data more repeatable, and we can identify anomalies in our data. So the data that you will have been given by Mr. Hancock and Mrs. Lees should look like this. So you can see, first of all, our clear headers. We have the independent variable normally goes in our first column, concentration of sucrose solution, and we've got the correct units, moles. It's just a capital M. We've got the initial mass of the chip, and they were all 2 grams to start with, because hopefully you cut them all to the same length. So it's one way to check that you are keeping your control variables the same. And then we have our end mass. And as you can see, some have gone up and some have gone down. So when we write the change in mass, we must make sure, so as not to confuse ourselves, that if it increased in mass, we put a plus, and if it decreased in mass, we put a minus. If it stayed exactly the same, you wouldn't need a plus or a minus. It would be 0.0. To work out percentage change then, so for this example, we would have had change in mass plus 1.5 divided by starting mass 2 times by 100, and that should give us 75%. So we calculate all of those for the data that we have, and then we're going to put these into a graph. So your graph is a little bit unusual to graphs you've normally drawn. Instead of having your x-axis that goes across down at the bottom, your x-axis would be somewhere in the middle of your y-axis. And this allows us to plot positive numbers above the 0, what we call the origin here, and negative numbers below the 0. So this, in effect, this y-axis is a little bit like a number line, going from negative numbers, okay, to 0, and then positive numbers up. And we've got percentage change in mass. Concentration of sucrose solution, you would choose your appropriate even scale along the middle here. So if you can come up with your own scale for your graph, and this would be on page 48 in your booklet to design your scale for your graph. So start somewhere near the middle of the graph for drawing your x-axis going across the page, and then you can choose an appropriate scale going upwards and downwards for your y-axis. So thinking about what the maximum increased change in mass needs to go up to and what the maximum decreased change in mass goes to. So here you can see an example of the scales. So we call here where the axes cross the origin, and it's always 0. So we need to go up in equal amounts. So because we need to go to 75 for our highest increase in mass, I've gone up to 80 here, doing every 10 little squares 10. So it's a really easy scale to work with. And coming down, our lowest decrease in, or highest decrease in mass is minus 50. So down here, so I've come down to minus 50 on this scale. And then you plot your points precisely, and you need to draw in your line of best fit. So it might have roughly even numbers either side of the line, or it might be going through the majority of points with perhaps one result that seems to be either a little bit high or a little bit low, what we call an anomalous result. So what do these results actually mean? Can we explain what this graph means? Whenever we're explaining the osmosis graph, I tend to split it up into about 4 kind of short paragraphs. I explain what's going on here at the top, that it's increasing mass, and it's increasing the most mass, and why is that? So think about the concentration. It's at 0 molar, which means it's in pure water. Then we can see as the increase in concentration, we get a decrease in mass, until when we get to 0.6 molar here, we've actually got now a decrease in mass, and it goes down the other side of the scale here into the negative decrease in mass part here. So we kind of split it up into describing what happens along this graph. So for example, at 0. molar pure water, the chips gained in mass the most. This is because, so this is the scientific explanation, there are more water molecules outside the chip than inside the sap vacuoles of the potato cells. The water moves down a concentration gradient from high water concentration to low into the sap vacuoles through the semi-permeable cell membranes by osmosis, causing those cells to swell and become turgid. And if you'd felt the chips that have been in the pure water, they would have felt quite firm and hard. So sometimes when we're making chips or frying at home, we'll soak the potatoes in water first to make them firmer, but we need to make sure that you dry them off, obviously, before you stick them in hot fat, because water and hot fat do not mix. At 0.2 and 0.4, they also gain mass for the same reason. However, there's a less steep concentration gradient. So less water will move into the cell, so the percentage increase is less, so it's starting to go down. At 0.6 though, the chips are now starting to lose mass, so water is moving in the opposite direction. And this is because there are more water molecules inside the chip than outside in the solution. So water moves down the concentration gradient from high to low, through the semi-permeable cell membrane in the potato cells, out and into the solution by osmosis, so going in the opposite direction. And again at 0.8 and 1 molar, they all lose mass for the same reason. However, as the concentration of the sucrose solution increases, this percentage decrease increases as the concentration gradient gets steeper. The important part in this graph, if I go back to the graph, where this graph crosses the x-axis, so if you can see there, if that's 0.4 and that's 0.6, halfway is 0.5, and halfway again is 0.55. So at that point there, that's a really important part in the graph, because where the line of best fit crosses the x-axis, so at approximately 0.55 molar, this is where the potato chips are not gaining any mass or losing any mass. So there's no net gain or loss of mass. And it's at this concentration that we say the concentration of the solution inside the vacuole of the potato cells is exactly the same. And we call that concentration, that's equilibrium. So no gain or loss in mass is equilibrium. So hopefully you should have completed the pages from 45 to 49. So page 47 is for you to write your conclusion down on. There's a little task on page 49 where, again, it's just a practice being able to calculate a percentage change, a positive and a negative one, and showing your workings in the example. A problem-solving task on a potato that's been put in some distilled water with strong solution in a little pit in the middle. So explain what's happening. And then pretty much a repeat of what you've just seen, but I want you to have a go yourself. So page 50, 51, and the top of page 52 is the same kind of experiment that's just giving you some different data and seeing if you can plot those graphs yourself. Thank you.